Good evening, everyone. We're going to go ahead and get started. My name is Eric Cole, and I'm the founder and president of the Dandy-Walker Alliance. We're excited to welcome you to the first ever Q&A webinar that we are hosting. Today, we're joined by Dr. Kimberly Aldinger, a principal investigator in the Center for Integrative Brain Research at the Seattle Children's Research Institute. Dr. Aldinger's lab studies how genes influence brain development and how those processes can malfunction to cause neurodevelopmental disorders. She's particularly interested in understanding disorders of the cerebellum, such as Dandy-Walker malformation. Dr. Aldinger was first introduced to Dandy-Walker as a graduate student and has since expanded her focus on it as a full-time researcher in Seattle. In 2019, she used whole exome sequencing to look for genetic causes of Dandy-Walker, and now she's working to sequence the full genome in the hopes of linking more genes with the malformation. Dr. Aldinger has generously agreed to take some time with us this evening to discuss her research and answer any questions you may have about Dandy-Walker genetics, common associated conditions, cerebellar anatomy, and more. But first, a few ground rules. Before we hand things off to Dr. Aldinger, here are a few things that I want to go over. Number one, there's a lot of you here, and even more who aren't here, but send in questions ahead of time. So we're going to record this webinar, and it will last an hour, and we'll try our best to answer all your questions in that time frame. But please understand that we may not be able to get to everyone's questions. Chris will address that more towards the end of the webinar about how to send additional information to us or questions in. Second, if you wish to ask a question live on the call, please use the raise hand feature at the bottom of your screen. And Chris, our executive director, who is going to moderate tonight, he will call on you. Alternatively, you can type your question in the Q&A section at the bottom of your screen as well. But we would ask that you stay muted unless Chris calls upon you to ask a question. Finally, we are recording this webinar and it will be posted on our YouTube channel next week. If you do not wish to be recorded, you may either stay silent during the call or hop off the call now and watch the recording when it's posted in a few days. I think that covers everything. So without further ado, I'd like to welcome Dr. Kimberly Aldinger. Dr. Aldinger, thank you so much for taking the time to be here with us today, sharing your research with us and answering some questions from our community members. So let's start off. Tell me a little bit about your Dandy-Walker genetic research.
Thank you very much for the opportunity and the invitation, and I'm happy to present today. Just one moment. Okay, I think we have success if we put it in presenter mode. Okay, I think you should now see my slides. I'm getting the thumbs up from Chris, so that's a good sign. So Eric provided a little bit of foreshadowing about what I'm going to talk about today. I'm going to talk about Dandy-Walker genetics in general, and a lot of it is based on the work that we were able to do just a few years ago. So what is Dandy-Walker malformation? We'll talk about that, even though most of the families and individuals who are watching today have a much more intimate knowledge about this than I may have even. I want everybody to be on the same page. We'll talk about the classic genetic evidence and then more recent genetic evidence. I'll provide a brief summary and then I'm happy to take your questions. So I really like to start everybody on the same page with what even is the cerebellum. It's a mouthful to say, and in this video on the right-hand side of the screen, you can see the cerebellum is at the back of the brain. This is an adult presentation of what the cerebellum looks like, and just to give localization as to where in the brain we're actually talking about, because there are many different places and spaces there. Okay, so the human cerebellum, despite looking like a very small size, has most of the neurons that are present in the brain and actually in the total body are located in the cerebellum. So 80% of all the brain cells that are in our bodies are in the cerebellum, and its structure, so the way it's built, the way it's put together, as well as what it does, is similar between mice and fish and humans and monkeys. So it's a really important structure for it to be working correctly in order to be able to do its jobs appropriately. And what those jobs are mostly classically known for motor coordination. So there's posture towards the middle of your body and posture from the outside of the body. Being able to walk on a straight line, for example, is related to having a functional cerebellum. But we know that the cerebellum is involved in a lot of different things also. So not just motor coordination and sensory processing, but also in cognition, emotion, and language. And so this diagram, which nobody's meant to understand really, but the point is that the cerebellum is this tan-shaped structure in the bottom right-hand side. And there are a lot of other different regions of the brain that have different things that they control, different functions that they control in the body. And the arrows and lines show how all of these different places in the body are connected. So we can see that the cerebellum, through a couple of connections, is very closely related and interacts with many different regions within the brain. And so, excuse me, if there is some injury or difference about the cerebellum in the way that it was formed or the way that it's working, that could have an impact on the way that it interacts and processes information from the rest of the brain too. So Dandy-Walker malformation, which is depicted on the right-hand side, is an imaging pattern. And so what this means is that by looking at this image on the right-hand side, which is an MRI image of the middle of the brain. So if we take a slice of the brain with imaging right down the middle, this is the picture that we see, sort of like opening up, cutting an apple in the middle and looking up the middle of the apple where the seeds are. So what we can see is that the cerebellum in Dandy-Walker, in order to make that diagnosis, the cerebellum vermis, so the middle part of the cerebellum, is small. Instead of being right next to the brainstem, which is this vertical structure in the middle with the big bump on the front hand side towards the face, that it should be right next to the cerebellum, and that the cerebellum should be right next to the brainstem. It's not, and then it's also turned up, it's upwardly rotated. So there's a big angle that's between the brainstem and the cerebellar vermis, and there's a fluid-filled space in the back. So those are the three criteria that are required to diagnose this imaging feature of Dandy-Walker. And there's some prevalence estimates about how frequently this occurs, and it's about 8 in 100,000 individuals. That's a difficult number to actually be accurate about, because in order to make that diagnosis, there needs to be an image of a brain image. You need to be able to look at the brain. And so that's either through an MRI, through an autopsy, are the two ways that can take place, but not everybody has that done. And so it's difficult to actually get an accurate accounting for how many individuals have Dandy-Walker malformation. Dandy-Walker malformation can also occur by itself without any other clinical features in an individual, or it can occur as a syndromic presentation. And so what that means to be syndromic with Dandy-Walker is that not only is this Dandy-Walker pattern in the brain, but there are other clinical features in that particular individual. So maybe cardiac change, maybe heart changes, maybe gut changes, maybe vision changes. So different parts of the body could also be affected, but they don't have to be. It can also just be the only diagnosis that's made for an individual. Excuse me. And so that it has a lot of value for what the outcome is going to be for a particular individual with a Dandy-Walker malformation diagnosis. So about half of the individuals who have a Dandy-Walker diagnosis have a favorable neurodevelopmental outcome, and half have some significant challenges. And whether or not those significant challenges will be present is really addressed by what the additional clinical features are that are present per individual. So as I already mentioned, there are some other symptoms that can be present. For example, epilepsy, autism, hydrocephalus is very common. Heart defects can be present, and there can be other brain or other body clinical features also. The inheritance most of the time is sporadic. That means random. That means typically it does not run in families, although there can be presentations of rare families that have more than one individual that have this diagnosis. And the recent estimates suggest that the genetic cause is the genetic risk is about 16% of individuals, which is a pretty low number. But I'm going to describe a little bit more why this is an underestimate of what the genetic risk actually is for individuals. There's also a risk for prenatal risk factors we know can also contribute to Dandy-Walker malformation, and that's about 30%. And I'll talk in a couple of slides, excuse me, about where that information comes from. And then as I mentioned, the recurrence risk is low, but that's in the cases where the presentation is sporadic, so just one individual in a family. It can occur in more than one individual in a family, and that's rare, but it can happen. And that's where genetics can play an important role too, to determine what the risk to families could be. So why do we think that Dandy-Walker could be genetic in the first place? Well, early on genetic evidence, and when I say early on, I mean the early 1900s through the 1980s, when individuals or clinicians would write about a description of what the clinical features were of a patient and publish it in a medical journal to be able to share that information with their colleagues. And so some of the information that suggests that Dandy-Walker malformation is likely to have a genetic cause is the timing of it. So we know that Dandy-Walker starts really early in pregnancy, and on the left-hand side, there's just a schematic of a first trimester pregnancy, and there are reports in the literature about first trimester miscarriages. Those miscarried fetuses did already have evidence of a Dandy-Walker malformation presence. And so that suggests that something that's happening as a birth defect, so really early in development, we know that 80% of all birth defects in general likely have a genetic cause. I just mentioned that it doesn't usually occur with siblings, but now I'm using that same piece of information to say that when it does occur in multiple siblings, that's also evidence of genetics. So there are not a lot of families. There are very few families that have multiple individuals that are depicted here just with the purple dots who have Dandy-Walker malformation, but it does occur. And when something happens more than one time in a family, that gives us evidence that there's a likely genetic cause for it. And then also early cases when we had the ability to look at chromosomes, an extra copy of a chromosome or loss of a copy of a chromosome. So in this case, I've depicted an extra copy of chromosome 9. Individuals who had these extra or missing pieces of chromosomes have also been diagnosed with Dandy-Walker malformation. And so that's adding or removing some pieces of genetic information that also gave us a clue that there is likely to be a genetic cause for Dandy-Walker malformation. But this is a lot of old information. And we really look at genetics in a different way nowadays. And this is thanks to next generation sequencing, which was really borne out by being able to sequence the human genome. So in 2001, we had the first human genome sequence, and the technological advances that allowed us to do that allow us to now do much more in-depth characterization of the genetic profile of an individual in general. And so we're just gathering more and more information every day as time marches on. So I want to talk about the three primary tests that are more standard nowadays. Some are clinically offered and some are still more in the research space. So genome sequencing is sequencing every bit of DNA that's present in an individual, excuse me, and that's really done more so on a research basis still. The cost and the time to do that is coming down, and so hopefully that will more commonly occur in the course of clinical genetic testing soon, but it's still not widely offered as a clinical test that is covered by insurance, the clincher sort of being the covered by insurance component of that. We do have exome sequencing, which can be offered clinically, and this sequences just 1% of the entire genome. So just the little coding pieces of information, the pieces of information that will lead to making a protein, and the protein is what the function of the gene does. So that is called exome sequencing, and we're able to do that on a clinical basis and often obtain excuse me, insurance payment, insurance authorization for that. And then there's targeted gene panels. So targeted gene panels require knowing what the genes are that you want to look at and you want to sequence in an individual. And that can be really helpful for some diagnostics. If you have a specific epilepsy, for example, sometimes that can be helpful, where we know that individuals with Dravet syndrome, for example, most often have a mutation in a gene called SCN1A. And so if we sequence SCN1A and a couple of other genes, we can most likely capture the information for those individuals. But when there are lots of genes to look at, it actually becomes economically favorable to do the exome sequencing rather than picking and choosing which genes we think might be related, because sometimes we make really good educated guesses and sometimes we're missing information by just looking at individual genes. But it's cheaper and faster. So that's the reason why sometimes targeted gene panels will still be done. So overall, there are a lot of benefits for attaining a genetic diagnosis in general. And primarily what's relevant for individuals with Dandy-Walker malformation is to secure clinical diagnosis when the symptoms of different clinical entities overlap. And I'm going to talk a little bit more about that in the next couple of slides. It also allows us to pinpoint the cause. So if we can identify what the genetic cause is, then we have a name and a potential candidate that can be used to access different treatments in the future. We can also predict symptoms and severity better by looking at individuals at different ages who have changes in the same gene. We can get an estimate as to what we expect the clinical course to look like for a particular individual. And that can be really helpful prognostic information. What symptoms do we expect to see? What symptoms are present now? What symptoms might occur as an individual proceeds through life? What sort of therapies might be more effective or useful or should be considered, and that leads into choosing the correct treatments. So there are some treatments that are indicated or should not be used when you have a particular genetic diagnosis, and so that can be much more helpful and much more targeted than taking a more sort of generic approach to a clinical disease. And then also accurate genetic counseling information is very important too, because as I mentioned, that most often Dandy-Walker malformation is sporadic, meaning typically one individual in a family. There are rare cases where there are families who have more than one family member affected. And so whether or not it doesn't matter if it's rare, if it's sporadic, or if it's inherited, unless it's your particular family, and then it matters a lot. And so having that important genetic counseling information is really valuable, which is what the underlying genetics can help with. And then early screening for treatable conditions. So we know that newborn screening is increasing nowadays. There are a lot of N of 1 trials that are beginning to emerge in treatments for genetic neurodevelopmental disorders. For example, just yesterday, there was a new treatment for Rett syndrome, which is a mutation in the MECP2 gene that was approved by the FDA for clinical trials. So I want to talk more about the clinical features, and I'm going to start with just the imaging diagnosis in general and about the confusion that happens a lot of time with diagnosis of Dandy-Walker malformation, or actually all cerebellar malformations. And so there are two categories of malformations that we recognize within the cerebellum, and some are a specific abnormality and others are a pattern. And so those with a specific abnormality, there are very few of those, and they're actually depicted on the left-hand side with some acronym letters, which I didn't put what the acronyms mean, but RES is rhombencephalosynapsis. And in that instance, there's no middle part of the cerebellum. The two halves are kind of joined together. And that's a very characteristic pattern or abnormality to recognize. And so we think that there are really strong genetic roots to that. And then the other is molar tooth malformation. I'm going to admit that when I first heard about the molar tooth malformation, I thought it had something to do with actual teeth, and it does not. It's because on this lower left-hand side, the middle part of the brainstem actually looks like a molar. And so that's why it was called the molar tooth malformation, with the roots and these two black arrows indicating the long roots of that the brainstem looking like this tooth presentation. Everybody who has this tooth looking shape of the brainstem has Joubert syndrome. Excuse me. It's called the molar tooth malformation, and they have Joubert syndrome. And it's really important to make that diagnosis. Joubert syndrome is a recessive condition. It has some surveillance that's important to consider, and about 40 different genes that we know are causal for Joubert syndrome. And so the specific treatment and prognosis is really tied to that information. And so it's really important to make that diagnosis. So if we move over to the right-hand side where the recognizable patterns of different hindbrain malformations, now I actually am showing you what a typical cerebellum should look like that's labeled with normal. I'm not sure if you can see can you see my cursor? You can. Okay, Eric's shaking his head. Okay, so I'll use it then. So in the normal pattern, we can see the size and shape of the cerebellum. This little black arrowhead, which might be a little difficult to see, is the fastigium. And then there are two folia, these arrowheads, which are indicated here. And then we have Dandy-Walker malformation, which we are already sort of talked about. But if there are questions, I can go into more detail during the Q&A as well. But so this is the middle part of the cerebellum that is away from the brainstem. It's at a 90-degree angle here, and there's a big fluid-filled space underneath and around the cerebellum. And then there's Blake's pouch cyst, excuse me, which can be really difficult to identify unless it's a trained neuroradiologist, actually, or some particular neurologist as well, to be able to actually identify the particular features of Blake's pouch cysts. The reason why it's important to make this diagnosis is because sometimes when it's diagnosed prenatally, it actually goes away when you look at the brain again later in development, and it actually has a good prognosis, meaning that the neurodevelopmental profile expected in individuals who have this presentation is very favorable. So not significant challenges or normal development. So it's important to make the distinction, but it's a hard distinction to make without being a trained neuroradiologist. And then megacisterna magna is MCM, and this is where the cerebellum is the normal size and shape, but there's a space underneath it. And then there's an arachnoid cyst, which sort of looks most similar to megacisterna magna, but you can see that there's actual physical pushing of the cyst. The cyst is filled with fluid, and so the black arrow is indicating that the middle of the cerebellum is actually smushed, and it's because the physical pressure from that cyst is physically pushing the cerebellum. So what we would expect is if that cyst were to go away or not be present anymore, that the cerebellum would sort of unsquish, sort of like if you take a beach ball and squish it and then let it go, and it forms back to its space. So this is a physical, imposes a physical change on the cerebellum that is not the same as not being formed correctly. And then cerebellar hypoplasia means that the whole cerebellum is small. It can be just the middle part, but it's more typically the whole cerebellum is small. And so those are some distinctions. But the genetics and the clinical features are quite variable with these different recognizable patterns. And so that can make prognosis and expectation for outcomes difficult, excuse me, to predict. So cerebellar malformations in general are frequently diagnosed prenatally. This is another reason why genetics can be really helpful, because if the genetics are identified at this early phase in development, there's the potential, if the genetics are known, to provide prognostic information for families at this really early phase. But the cerebellum, as it's developing, so it's the second most having a small cerebellum is the second most common reason that a pregnant woman will be recommended for a fetal MRI. And sometimes that more sophisticated view of the fetus's brain will determine that it was a false positive and it looks okay, or there might still be concern for how the development is happening. And then, as we know, as we sort of just talked about, then going from this prenatal state, some of these prenatal features of what later very different looking and presenting cerebellar abnormalities or syndromes might look like can look the same really early in development. And that's a function of the cerebellum's only developed so far, so we can only define it in so many ways. And so depending upon the time that the abnormality is found and looked at in more detail, we may or may not be able to make better prognostic information or expect what that's going to look like, because the cerebellum hasn't developed enough for us to see how it looks different than it should. Is it just small? Just small later on, as the cerebellum grows, can change a little bit into what its presentation looks like. But this is also a motivating factor for seeking genetic information, because without that, we're just left to looking at the clinical symptoms of an individual as they grow. And then we receive the very frustrating advice from physicians that says, oh, let's wait and see. Oh, we didn't quite make this milestone, but there's variation in how everybody attains the time to walk or to talk or to crawl. And so that can be very frustrating. I speak from experience as a child who has a different developmental challenge, but it's very frustrating to have the wait and see sort of expectation from clinicians. So genetic information can help alleviate a lot of that stress and uncertainty if we're able to attain it. So prior to this analysis that we did a few years ago, there had been no systematic analysis of patients with Dandy-Walker malformation on a genetic basis previously. So it was mostly case reports. So individual patients who were identified that had mostly a chromosomal change or maybe a single gene change were reported in the medical literature, but nobody had looked at a group of patients with Dandy-Walker malformation and specifically asked the question about, are there genetic changes that we can see in these individuals? So that's what we sought out to do with the support of the community of the Dandy-Walker Alliance. So I want to thank everybody who did participate in this effort and be able to share that information back now. Excuse me. So our two primary questions were, what are the genes that are associated? And are Dandy-Walker malformation and cerebellar hypoplasia, are they separate things? Are they biologically different, even though they're happening in the same place in the brain? Or is there a genetic relationship to them? So to do this, we looked at individuals who either had cerebellar hypoplasia, basically individuals who did or did not have Dandy-Walker malformation. So either cerebellar hypoplasia, which we know there are a lot of genetic syndromes, single gene disorders that have cerebellar hypoplasia as one component of their syndrome, and then individuals with Dandy-Walker malformation. The individuals, excuse me, with Dandy-Walker malformation did include those that had syndromic presentation, so other clinical features, and individuals who had only Dandy-Walker and not and who were able to attend university, who were able to lead, a productive life, and did not have other significant neurodevelopmental or body system concerns. And so we use trio sequencing for this. Mostly what that means is we had DNA from both the patient as well as the unaffected parents. And so this allows us to make the comparison of what is present in the child that is either absent from the parents, or since the parents didn't have the condition that we were looking at, they might have, in a recessive case, one broken copy of a gene where the child ended up with two broken copies, but you need two broken copies in order for the symptoms to appear. So this is the how we looked at these data, it was about half and half of the 100 families with cerebellar hypoplasia and Dandy-Walker. And then we performed exome sequencing. And through that analysis, we were able to determine that 36 patients had a single gene disorder genetic cause for their condition. And this resulted in 41 pathogenic or likely pathogenic variants in 27 genes. That's just a lot of numbers and letters that says there were 36 diagnoses, but 27 different genes. And so what that means is there were very few genes that we saw show up more than one time. There were 7 genes actually that we saw show up more than one time in our analysis, and it was usually only in 2 individuals. And so that means that for a cerebellar condition in general, there are a lot of different genes that are involved in development of this structure and leading to different symptomatic relationships. So if we sort of summarize these data into whether or not we identified a genetic cause or not, so among those 36 patients, most of them had cerebellar hypoplasia and very few had Dandy-Walker malformation. The individuals that we did identify a genetic cause for who had Dandy-Walker malformation did have other clinical features that indicated it was a syndromic presentation. So what we also did is, since there were so many genes, and a lot of these genes were already well described or known, we could then ascertain another group of individuals by their genetic cause. So instead of looking at a group of individuals with Dandy-Walker or cerebellar hypoplasia and looking for genes, we could then find individuals who have a genetic cause in one of those genes that we found, look at more individuals, and then look at their brain imaging and say, do they or do they not have a cerebellar change? And so in most cases, we were able to identify for those 27 genes that we identified that there were many additional cases of individuals who had a change in that particular gene and also had a cerebellar malformation, but we rarely found Dandy-Walker malformation again. And so that's also questioning whether or not there's something sort of special or unique about Dandy-Walker malformation, or is it because that the test that we're using is actually very limited and we weren't able to capture the genetic changes that might be present. So another piece of information which is really valuable and speaks to the syndromic relationship, this is just a bar graph that is showing the same information that was in the table on the left side, where with cerebellar hypoplasia, 50% of those individuals, which is a really high it's a high diagnostic rate for identifying that there's a genetic cause, which suggests an individual with cerebellar hypoplasia and other features should seek genetic testing and likely find a genetic diagnosis, but that wasn't the case with Dandy-Walker malformation. And if we instead separated the patients by intellectual disability versus not intellectual disability, we can see that we did a really good job making a genetic diagnosis in individuals who have intellectual disability and not among those who did not. And so, as I mentioned, with Dandy-Walker malformation, we had more cases than you might expect in the literature of individuals who had isolated Dandy-Walker malformation, who would not have had intellectual disability. And so with cerebellar hypoplasia, we know that there's a really strong relationship between cerebellar hypoplasia and intellectual disability as it is. And that was really what we were identifying with some of these genetic causes. So I want to talk about, I'm talking about what the genetics of Dandy-Walker malformation are, and I just told you that we didn't find very much. And so why do we think that that's the case? Is that because we don't think that it's genetic, or is that because there are limitations in the testing that we're using? And there are a number of limitations in the testing that we're using. When I started, I mentioned that we used to look for chromosomal abnormalities. So whether or not a giant piece of genetic information was missing or there was too much. And so we're using much more sensitive methods now to detect genetic changes, but there's still more room to go. So this apple schematic on the bottom left-hand side is depicting what the exome actually represents. It's one tiny little slice of the entire genome. We still have a lot of information that we haven't surveyed. And there are different types of genetic information that are also not well captured by exome sequencing. For example, the mutation that causes Fragile X syndrome or the mutation that causes Huntington syndrome, those types of mutations, which are called an expansion repeat, when there's repetitive information, it's harder to detect with these methods. And it's very much harder with exome sequencing, excuse me. So there could be types of genetic variation. There are likely types of genetic variation that we missed because of the test that we use. So the test matters. Also, when and where we are in the space of time matters. So today, there's a lot more information and a lot more genes that are known to have a relationship, a genetic relationship to human disease than there were last year, than there were 5 years ago, than there were 10 years ago. So for any individual who has had exome sequencing in the past, excuse me, probably from 2015 forward and had a negative result, it's worthwhile repeating the analysis. So not the whole test, not taking blood again and getting DNA and repeating the analysis, but just comparing the results to the larger amount of information that's available. Are there more genes there are many more genes that we're aware of now that are associated with neurodevelopmental disorders or autism or epilepsy or other conditions as well. And so it's worth reviewing because clinical testing will only report out genetic changes that have been linked to your specific condition. And so if that's not the case, then they might list it as a variant of unsignificant VUS, variant of uncertain significance, or it might not show up on the test, which looks like the test is negative, but it's because a lack of information. It's not because a lack of detection. And then the limits are that, it's also, as I mentioned already, it's really important to have accurate genetic counseling, and it can be a prerequisite for gene-specific treatments that are coming online much more frequently nowadays, where there's a lot of effort to do that. So it's really easy to say that, well, since we didn't find anything with the exome, that maybe we just need to look harder. But we took a different approach with looking at the analysis as well. And this is because we know that there are prenatal brain injuries that are risk factors for having a smaller cerebellum. And so was that contributing to why we weren't detecting genetic causes for a large amount of individuals with Dandy-Walker malformation? Some of these risk factors that I'm talking about are hemorrhage or preterm birth or twinning. The human body is really only designed for one baby, not more than one, and specific brain features. So we could actually go back and look at the MRIs of every individual and determine whether or not they had some specific features in their brain that would suggest there was an injury. These things often mean asymmetry, so one side more effective than the other. That suggests that something happened to that one side versus the other. And so we wanted to know whether or not we weren't capturing genetic results because there was a higher risk of prenatal brain injury, and that's not what we found. So summarized in this bar graph are the entire group of individuals, and then separated by cerebellar hypoplasia or Dandy-Walker malformation. And the 30% in red in bottom is just indicating that among individuals where we did not find a genetic cause, but we did find evidence of of at least one of these items on the left-hand side, that there was evidence for an injury, that occurred in about 30% of individuals. So it could be that for 30% of individuals, you might expect a negative genetic result if there is evidence of having some of these prenatal brain injury. But then, were there any cases where we found both a gene and evidence of injury? Very few, and not any in the Dandy-Walker cases. And then the numbers in the diagonal blue and white stripes are the numbers that we identified in our cohort where we did find a positive genetic result. And so what's left over is that there are about half of the individuals with Dandy-Walker malformation that we didn't find evidence of a brain injury, and we didn't find evidence by exome sequencing of a genetic cause. But that suggests that it's a limitation of the test that we used, since these individuals also did not have evidence that they had a brain injury. And so that was really the motivation for starting to look at whole genome sequencing, because then that opens up the 99% of the genome that we haven't looked at together. That provides different challenges because there's a lot more information to interpret and to gather. So again, it becomes an information challenge, but it also provides an opportunity to look at those missing types of variation that we weren't able to capture with exome sequencing, as well as look at a lot more space in the genome. And those preliminary efforts are ongoing. So overall, Dandy-Walker can happen all by itself. It can be diagnosed alone or with other clinical symptoms, but it's just one piece of the puzzle to an individual's clinical presentation. And so looking at what the totality of symptoms are for an individual is really important for determining prognosis. And Dandy-Walker can result from an early brain injury, but our evidence is suggesting that it still is more likely genetic, which is why when I started, I said, we think that what we found is 16% of individuals, and why we think that that's really an underestimate of what the genetic relationship is. And then exome testing can be diagnostic, but mostly in syndromic cases, excuse me, of Dandy-Walker malformation. And exome really is a first-line test for individuals with significant neurodevelopmental challenges, for example, intellectual disability. And with that, I'm happy to take any questions you may have.
Dr. Aldinger, thank you so much. That is a lot of information, and I'm gonna have to go back and watch it again to absorb it all. One of the questions I had, and then we will open it up to the rest of the audience. But at the beginning of the presentation, we heard a little bit about many times in family we'll have one individual if they get diagnosed with Dandy-Walker malformation, but sometimes we will have more than one individual diagnosed with Dandy-Walker malformation. When we have a family where the diagnosis is present in more than one individual, as a scientist, is that easier for you to study if we get saliva or blood samples from them? It can do the whole genome sequencing or whole exome sequencing when it occurs more than once in a family, or does it not matter?
It can be easier, yes. And the reason for that is because because you're comparing what's present in those multiple family members. You know that it has to be present in multiple family members. And so that more information is better in that scenario. So a lot of times how we identify a new gene is related to a human condition is you have multiple affected individual. You might have 5 individuals and they could be from 5 different families, and that suggests there's real information there. You could also have 5 individuals from the same family, and that would mean that there could be an important genetic cause for that particular family that might be different from other genetic causes that we know about already. So it's very helpful to have that information.
Yeah, thank you, Eric, and thank you, Dr. Aldinger, for such a wonderful and informative presentation. I'm sure some of the people on the call here have some questions that they'll want to ask. I also have some questions that were sent in ahead of time, so I'm going to try and mix those in as well. So I'll start out with one that was submitted ahead of time, to give people a chance to formulate some of their own questions on here. But how does having genetic testing done, how can that help doctors find out how to better treat issues associated with Dandy-Walker, if at all?
So I think that there's most of the time in medicine, symptoms are treated, right? So we frequently don't treat, or clinicians don't treat the underlying genetic cause, but they treat the symptoms that emerge. So for example, seizures are treated with anti-epileptic drugs or sometimes surgery, or VNS, or there are some different strategies to use. Some of the strategies to use for specific symptom management can be informed by what the underlying genetics are. And so instead of trying treatment A and then B and then C, it might be already known that for that particular genetic cause, you go and treat using item C and not A, or not waste your time with trialing A or B. So that's where there can be helpful information. There can also be really helpful prognostic information if there are already a number of individuals who have that particular genetic cause, and looking at what their symptom course looked like, how symptoms changed over time as the individuals aged, or what treatments were effective, can have very important and helpful information for physicians and families.
Thank you. And I realize I didn't say this off the top, but there are two different ways to ask questions on here. We've gotten a couple submitted already. You can either use the raise hand feature at the bottom, or you can type it out in the question and answer section as well. Either one is good. So moving along, is there a way to determine before birth or from an initial MRI after birth how severe a case of Dandy-Walker is going to be, and how severe the co-occurring conditions might be?
So that's going to be based on what the symptoms are. So if additional abnormalities are identified prenatally, then that is most likely going to be a syndromic presentation. And we know that syndromic presentations have more significant clinical challenges throughout life. The challenge with actually answering this question is that there are some individuals who, when faced with a really difficult challenge of being diagnosed with a fetus with Dandy-Walker malformation or another birth defect, some individuals choose to discontinue with the pregnancy. And so then we don't have information about what would have happened had that child been born. So it's a very challenging question to actually address. Also because there are not a lot of outcome studies that have been performed to know what happens to the individuals more rigorously with who have been diagnosed with Dandy-Walker malformation or related conditions, and what the prognosis and expectations are. So I think that there's a real need in that space to examine that further, to provide that helpful prognostic information.
That makes sense. Thank you. And so moving along, there's a couple of questions that are fairly related to what you just presented on. There's one person who's asked, he has a sister and a son who were diagnosed with Dandy-Walker malformation. Should they be doing genetic analysis and counseling based on that? Whether for his own son and daughter, to see if that's going to be passed down to potential kids. Like, what, I guess a better way to frame the question is, if there's multiple incidences or even just one incidence of Dandy-Walker in a family, is it worth doing the genetic counseling going forward?
Yes, I think anytime there is a more rare condition, so something that doesn't happen all the time, or something that happens really early in development, it can be very beneficial to pursue genetic counseling and meet with a geneticist to get that accurate prognostic information as much as you can for not only the current family members, but for additional or future family members as well. So if there are multiple family members that are diagnosed with Dandy-Walker malformation and those families have not received genetic counseling, I would strongly recommend that you pursue genetic counseling.
And is Dandy-Walker progressive? Like, does it suddenly show up? Are people who are diagnosed with it later in life, did that change over time, or was it always present and they were just unaware of it?
I think that this is a really common and important question. And Dandy-Walker malformation is a birth defect. And so, as I mentioned, we know that it starts during first trimester. So very early in development. You may or may not know that you have it unless you have a reason to take a picture of your brain. And so that's really the only way to be definitive and to know that change in the brain is present. And that also means that that's one of the challenges why we don't really know exactly how many people there are who have this particular brain feature and what the prognosis is. If we identified individuals who did recognize at 40 years old or 60 years old that they had Dandy-Walker malformation, what happened through the course of their life could really provide helpful information to younger families and individuals as well. And so, no, if it does not show up, it does not progress, it does not change. Once it's there. You just may or may not know it's there. There are other clinical features where that may be the case also, that are in the hindbrain that are until there's some symptom that would lead a person or a doctor to recommend an MRI or some kind of brain imaging, it wouldn't be possible to know if Dandy-Walker malformation were present.
Thank you. That's a great answer, and I think is going to be beneficial to a lot of people tuning in live and watching afterward, who may be diagnosed later in life and have a lot of concerns. So thank you for answering that. So we talked about the genetics of Dandy-Walker, but obviously there's a lot of co-occurring conditions that happen with Dandy-Walker, and that's where a lot of the symptoms come from, whether it's hydrocephalus or seizures, mobility issues, developmental delays. Have you found any genetic component with, let's say someone has Dandy-Walker and hydrocephalus? Is there a genetic tie that would make one more predisposed to having hydrocephalus than another?
I think that's a really good question, and we don't currently know the answer to that. So there is a really high co-occurrence of Dandy-Walker malformation and hydrocephalus. There are some large genetic studies that are taking place among individuals who have been diagnosed with hydrocephalus and finding some new genetic relationships there, but the individuals that were in those studies did not include individuals with Dandy-Walker malformation. So we don't have a good, I don't have a good answer to that question, but it's an important one.
Thank you. And obviously there's a lot of unknowns still with Dandy-Walker, and that's why the research that you're doing is so important. And one of the reasons that we want to keep supporting research, from you and from others in this field. So I think we've got time for a few more questions. There's quite a few that are in the queue here. So just, as a heads up, if we don't get to your question, I will read off an email address for you to send them to at the end that, we will try and answer after the fact. But we have a question here, a couple questions, asking about any ongoing studies that families could participate in. And what that might look like. So do you have any insight there?
We are not currently enrolling patients at this time. The way that research often happens is we evaluate many patients at the DNA of many patients at the same time. And so it's very different than a clinical scenario where one individual may go to the doctor's office and they may get recommendation for genetic testing or for an echo or some other test that's relevant for that particular individual. And in the past, we have collected samples and information from families, and then we wait until we have enough to sequence many at the same time. And we're in the process, we're still finalizing our initial genome sequencing analysis and have postponed enrolling additional patients for the time being until we can, finish that analysis. But you and Eric may know of additional studies for different aspects of Dandy-Walker that might be available to families.
Well, I think it's an outstanding question. I'm excited to see them coming in and asking it. And what I would say is please contact us and get a hold of Chris and let us get your information, because we will be doing this again and whatnot. As Dr. Aldinger said, when you think about doing genetic testing, we need to have a research coordinator that can help receive inquiries from individuals and families that want to participate this. And so when she was speaking earlier about collecting samples in trios from mom, dad, and affected child, well, somebody's got to send the consents out. Somebody's got to receive all the information back, log it in. Then there's staff in the lab that have to extract the DNA and whatnot, and then it gets sent off for the sequencing, and that comes back and the analysis is done. As Dr. Aldinger said, it's much more cost effective to do that in batches than it is to do it, just dribs and drabs. And so we will want your information. We're excited that you want to participate. One of the challenges many times in doing this is in following up with people to make sure that they follow through and sign the consents and send the samples. People that send the samples and don't send the consents back, there's not a whole lot there's nothing that can be done from an ethical standpoint in that. So there's a lot of communication and coordination involved in it. So again, we will do it again. The Dandy-Walker Alliance is committed to continuing to fund this with Dr. Aldinger's lab and whatnot. So stay tuned and we will get back in touch when that opens back up.
Yeah, thank you, Eric. And thank you, Dr. Aldinger. I know that being able to do clinical research on Dandy-Walker in a number of different capacities, clinical, genetic, can really be so beneficial. And I think your presentation tonight has opened a lot of doors that we can go down in terms of where we want to look next. But there's so much more that we still need to discover. Dandy-Walker wise. I think, Eric, you were going to jump in there.
Yeah, I have another question. And having a son with Dandy-Walker, Dr. Aldinger, what are the chances that if he decides to have a child of his own one day, that his child is going to have it? My grandchild then, I think, do they call it risk of recurrence, or how does that work? And do we know enough to know the likelihood of him passing that along to his son or daughter?
We do know that answer in the cases where there's when there's a genetic cause that's known. So one of the things I didn't mention, but I'm going to mention now because I think it's really important to remember, is that things can be genetic and not inherited. And that, typically we think of things that are genetic running in families. If an individual with Dandy-Walker, it's really important to meet again with the genetic counselor to have the appropriate information. So if there's a genetic diagnosis, it depends upon how that genetic diagnosis presented in the individual. So if the individual had a de novo change, meaning it wasn't present in their parents, but it was present in the child, and that child has Dandy-Walker, then what are you concerned about transmitting? Is it the Dandy-Walker, or is it that genetic change which might have Dandy-Walker or other things also. And so if it's de novo, then it's 50-50, that an individual will, that the subsequent generation will have that genetic change. But then there's also the opportunity for pre-implantation genetic diagnosis if individuals are interested in pursuing that route. And it really depends upon what the inheritance pattern is for genetics that are known. It's a more challenging assessment to make in the absence of genetic information, clear genetic diagnostic information. But it's really important information to discuss with the genetic counselor.
I think we've got time for one more question that I'll read off here. Is there a place where families can find more information on specific research findings or recommendations, especially if they have another co-occurring condition or disorder? The question here is specifically about trisomy 18, but this could go for other disorders as well.
I think that there are a lot of patient advocacy groups now. So there's a Dandy-Walker Alliance, which is extraordinary for individuals with Dandy-Walker malformation. But there is also, for example, trisomy 18 advocacy groups also. And so that might give a different perspective on different symptoms. And I would encourage individuals who might fit into different spaces to investigate each of those spaces and see where they think they might fit best or might find the most helpful information or resources. A lot of time that resource information crosses boundaries and diagnoses. So individuals who use a G-tube or use a wheelchair can cross many different boundaries, or individuals who have behavioral issues, different treatments and therapies may work, and parents are really the driving force, I think, behind sharing a lot of that information, and it's really invaluable to connect in that space. So I would encourage individuals who can align with multiple different groups to see which groups might align best, or even if multiple groups might be really beneficial to participate in or learn from or share information with.
Thank you. That's a great answer. And I think, like you said, so many of these things are interconnected and there's a lot of crossover in it. So I think the more that we can promote finding the information that's specific to your condition or your child's condition, I think it's so beneficial. Eric, do you have any other questions for Dr. Aldinger while we have her?
Well, I just want to say thank you, Dr. Aldinger, for taking time out of your Saturday afternoon there on the West Coast and spending it with us and sharing with us. We look forward to continuing to learn from you, to learn from the science, and supporting your research in your lab going forward from the Dandy-Walker Alliance perspective. So thank you for all that you've done for our community. So Chris, why don't you wrap us up here?
Yeah, thank you, Eric. And again, thank you, Dr. Aldinger, for spending some time to answer questions and share some of your work, and for all the work that you guys are doing out in Seattle to better understand Dandy-Walker and the causes behind it. Obviously, we didn't have a chance to get to everyone's questions tonight. I wish we did. If there are unanswered questions that you'd like some more insight on, please send them my way to awareness@dandy-walker.org, and we will do our best to answer them or at least point you in the right direction. We are planning to do another webinar coming up with a clinician. So if you have some more clinical focused questions that didn't get covered tonight, we'll send out some information on how to register for that, hopefully in the near future, and can get some of those answered then as well. But again, thank you, Dr. Aldinger, so much for taking the time to chat tonight and share so much of your work with us.
Thank you very much for inviting me to do this and being able to share what we found so far with the community and the families who are really integral to really lead it. You know, I'm just a steward and a facilitator, and really it's the families who are behind all of this work.
Yeah. Well, thank you again. And thank you to all of the everyone who tuned in tonight. I know we've got people from all across the US. I know there were some people from the UK and around the world tuning in tonight. So I really appreciate you all participating and asking questions, and being a part of this, and for all that you do in our Dandy-Walker community as a whole. So thank you to everyone for participating and showing up tonight. And we look forward to the next one, and look forward to all of the continued work that Dr. Aldinger and her team are doing. So thank you so much, and have a good night.